Okay, in the last section we talked about the organelles of the cell, and in this section we're going to talk about some of the structural portions of the cell. In the cytoplasm, this is really kind of the entire interior of the cell minus the organelles. So what you have is the microtubules, you have the microfilaments, the intermediate fibers. This makes up the cytoskeleton, and then you have all of the cytoplasm or cellular fluid. And then when you mix in with that the different organelles, this is where you kind of get this entire network of cytoplasm. So the cytoplasm includes all of the microtubules, microfilaments, intermediate fibers, the organelles, and the intracellular fluid, or the cytosol as it's called. And what the cytoplasm basically does, along with the cytoskeleton, is it provides this structural support for the cell, because animal cells, of course, have no cell walls. And the cytoplasm, the cytoskeleton rather, is the anchor point for a lot of these organelles to kind of just keep them from basically moving around the cell aimlessly, more or less. They're kind of anchored in place. And the cytoskeleton also allows for kind of transport of different types of things through the cell. And we watched a video in class of this entire process. Okay. So the cytoskeleton, this is that support network we said that consists of those different types of fibers. And if you look here, you can see the what we have right here is kind of an electron micrograph of microtubules and microfilaments, so you can kind of see how these things are organized. And with the really with these structures, it almost looks like a screen. Okay, so if you look through a screen, and then contained within these hollow parts, this is where you're going to find the cytosol, or the liquidy interior with the dissolved solutes and things like that. And then within these kind of webbed screened area, this is where you would find these organelles kind of attached to them. Okay, so now microtubules, these are the large structures found within the cell, and they consist of alpha and beta tubulin proteins. And what you have is, if you're going to move a vesicle throughout the cell, you have these kind of motor proteins that are powered by ATP, and they basically kind of grab the microtubule almost like it's a rope and kind of walk themselves along the microtubule, and that's how these vesicles move kind of throughout the body. Okay, and I had a professor in college who studied this sort of thing. And basically, for instance, if this is a neurotransmitter here, and it's being synthesized in the cell body of a neuron and kind of moving down the axon really to the spot where you're going to discharge it from the cell, that would be one example of this type of thing that you would see this movement from point A to point B a long way through. And there's many other examples too, but that's just kind of a common one. Now the microtubules themselves, these make up the cilia and flagella of the eukaryotic types of cells. And they have a very unique arrangement. It's called a 9x2 arrangement here. And basically what you have is you've got a series of doublets. So here you can look, and if you were to count all these all the way around, you would see 9 of them wrapped around this kind of central core here of 2. So there's your 9x2 arrangement. And what that basically allows for is these things like the cilia and the flagella, when they're moving in these eukaryotic cells, to kind of be able to bend back and forth across one another. Okay, and they're different than these flagella are different in bacterial types of cells. It's more of like a rotor, like a kind of like a stand mixer with a dough hook on it or something like that. That would be a prokaryotic type of a flagella, whereas the eukaryotes are arranged in this 9x2 arrangement. And when you get down kind of down here towards the kind of base of where they're attached to the cell, the arrangements are in these little triplets, which is a little bit different, kind of like the spokes attached to this central part here, and it almost looks like spokes on a bike wheel. So those are a little different. It's the same structure, it's just where you're looking at it within the structure. Again, down here, this is the attachment point in the cell, and then this over here on the left, this is up a little bit higher. And these are kind of unique for the eukaryotic microtubules. Okay, if you take an electron micrograph of a ciliated epithelial type of a cell, this is where you've got these cilia that are going to say beat and get the trapped mucus and things like that out of them. If you were to cut this cell kind of along this type of a plane and then take a picture of it right here, this is kind of what it would look like. And you can see again the 9x2 arrangement. You've got these 9 doublets all the way around the outside edge if you were to count them, and then you've got this central doublet in the middle. So there's your 9x2 arrangement in the cilia of this epithelial cell. Okay. Now microfilaments, micro as the name suggests, are the smallest fibers, and these are built from actin protein, and these provide structural support and are not involved in any motility or movement or anything. And what they're going to work with, for instance, in muscle cells, they work closely with myosin, and they allow for contraction. Okay, so I think I just said they're not involved in motility, and I misspoke. I'm sorry. They are involved in motility, as far as like the movement of the muscle cells, the movement of the fibers within the muscles, which we'll talk about later on in the course when we talk about the human body stuff, okay? And the structural role of these microfilaments is, for instance, in your intestinal cells, the ileum and the jejunum are ciliated. They're consist of villi and microvilli, which are greatly increasing the surface area of these structures because they're primed for the absorption of the nutrients that we're taking in when we're eating. And these tiny microvilli are these tiny kind of finger-like projections. If this would be, say, a villus, and then these are the tiny microvilli, you can see that if you just had like a flat structure, you've got some surface area, but as soon as you add a structure that looks like this, you've got a large amount of surface area for the absorption of these nutrients. And these microvilli have lots of little tiny extensions in them, and within them you've got a bunch of tiny microfilaments. So for instance, if you're looking through this type of a structure right here, there's a lot of microfilaments in there, and then they're attached down to the bottom here to these intermediate fibers. They're anchored to this kind of these intermediate fibers of the cell, and that's what's kind of giving them some of their structural support, allowing them to stand up and essentially do the job that they're primed to do. Okay, so we said microfilaments are involved in muscle contraction, but they're also involved in moving. For instance, like an amoeba, you can build up and break these things down very quickly and extend these pseudopods out for amoeboid types of movement. And then when we look at certain things under the microscope with the chloroplasts, what we're going to be able to see is some cytoplasmic streaming, where when you provide these lights, you're going to be able to get these chloroplasts kind of streaming around the plant cell and so forth. And the microfilaments are largely involved in the cytoplasmic streaming. So when we see it in the class, I'll be sure to point it out to everybody when we're doing the lab. The intermediate filaments now, these provide more of a permanent structural support. So the microtubules and microfilaments can be built up and broken down as needed, and the intermediate fibers, those can't. Those provide more of a permanent type of structural support for the cell. Now, plant cells, these contain the plasma membrane along with the cell wall. And what the cell wall does now is it provides some structural support for the plant. And the cell wall is made out of the cellulose, of course, that we were talking about. And it's indigestible, so we refer to it often as dietary fiber and things like that. And the young plants that are being built secrete this primary cell wall, which is very rich in polysaccharide. And then this is what's going to kind of glue these cells together to start providing some strong, integrity for the plant so that it can withstand, different types of environmental conditions and things like that. And then plants that live from year to year make a secondary cell wall, which is very strong. It gets lignified, and an example of this would be wood. So there's a lot of other types of things that can get laid down that really kind of add to the structural integrity of these primary cell walls that are being laid down. The plant cells also store carbohydrates as starch, in addition to the cellulose that's there, but they also store it as starch, which we've talked about. Okay. The animal cells, by contrast, they don't have any type of cell wall, but they have this extracellular matrix, which consists of glycoproteins like collagen, some proteoglycans, which you'd find, say, in cartilage or something like that. And they only have this plasma membrane, so no cell wall, so they need to make this extracellular matrix to kind of give the cell itself and the structure that the cells are making up, give it some integrity. And recall, animal cells, in addition to this extracellular matrix, they can store some of these carbohydrates as glycogen, but like we've said, previously when we were talking about the organelles, they don't have any vacuoles. Vacuoles are common to the plant cell. So if an animal is going to store anything, it's going to have to store it as really some sort of a biomolecule. It can't store it in a dissolved sense, like in a central vacuole or something like that. Now, as far as the junctions go that join the cells together, you have these intracellular junctions, and what they do is they allow cells to kind of communicate with one another. So what plant cells have are these little kind of tunnels that kind of connect the cytoplasm of the cell called plasmodesmata, and we'll see a picture of them here in a minute. In animal cells, the intracellular junctions, they've got a couple of them. They've got some tight junctions, they've got gap junctions, and they have desmosomes. Okay, so the plasmodesmata, as you look right here, for instance, here's 3 of them, and what they're doing is they're connecting, say, the cytoplasm of cell A with that of cell B. So they're basically allowing the solutes to move within the cell from cell to cell, and they make the cytoplasm of various cells kind of continuous with one another. Okay, so this, you know, you have 2 cells there, and you can see how they're joined together. Now, in animal cells, you'll have some tight junctions, and these are some continuous seals between adjacent cells. So what they're doing is they're preventing the contents of one cell from mixing with another cell. Other cells, or other tissues, I should say, are kind of linked together, and they're very strong out of desmosomes. And you can think of these as like rivets, or these strong kind of connections that are holding the 2 cell types together. And then gap junctions are very similar to the plasmodesmata that you would find in plants. The gap junctions are found in animal cells, and these are membrane proteins that are basically providing a junction between the 2 cells, and again, they're allowing for many of the contents of the cytoplasm to get from one cell to the next. So if we look here at this particular type of an animal cell, you can see that you've got a variety of the things that we just talked about. Up here, for instance, this line right here, these are tight junctions. So what this is doing is it's preventing the contents from cell A from getting into cell B. Okay, so it's going to stop the flow. You've got some desmosomes, so you can see here the extensions from this are kind of getting into that cell. The extensions from this one are kind of getting into that cell, as well as on both sides here and here. So what they're doing is they're kind of anchoring these cells together, giving them some really strong structural integrity. And then down here you can see the gap junctions, very similar to the plasmodesmata. This is say cell A, this is cell B, and then these are the contents. These are the little channels that the stuff can get back and forth between cell A and cell B. Okay, all right, and then some of the extracellular components include certain things like the cell wall, glycoproteins, proteoglycans, the extracellular matrix, and there's a handout on OneNote that I want you guys to look at from chapter 6, and you can read it, and it talks all about these extracellular components. And what these things basically do is they provide some supporting functions and help coordinate a lot of these cellular types of activities. Okay, so make sure that you take a look at that. And of course, as always, if you have questions with any of the things that I've talked about in here, please make sure you see me in class, and we'll go over it and make sure that you're all set with your understanding. Okay, so we'll talk to you soon.